Hello students, welcome to Simplify Pharma. Please do subscribe to my channel to stay updated with the latest videos. We are going to start with a topic, potential target diseases for gene therapy. These are the several topics that are going to be covered under this topic. The target cells of gene therapy and examples of several potential target diseases for gene therapy. Now, for the convenience of the students, I have divided this topic in two parts. In part 1, we are going to cover the target cells of gene therapy and with 3 examples of potential target diseases for gene therapy: type 1 diabetes, x-scid and cystic fibrosis in detail. Now, the target cells of gene therapy. When we talk about gene therapy, there are 3 main types of cells that we usually target using gene therapy: the hematopoietic cells, the hepatocytes, and the muscle cells. Now, the hematopoietic cells, they are most consistent target for gene therapy, has strong potential, whereas the hepatocytes, the attractive target for gene therapy because the liver plays a vital role in most metabolic process. Whereas the muscle cells, they are strong potential, they are used as an efficient target organ for correlation of intrinsic muscle and non-muscle diseases. Now, gene therapy has the potential to target and treat a number of diseases such as type 1 diabetes, x-scid, cystic fibrosis, Parkinson's disease, adenosine deaminase deficiency, hemophilia, blindness, and cancer. Let's start with the very first example, that is diabetes, in detail. Now, we all are aware of this. Diabetes is a disease that occurs when your blood glucose, also called as blood sugar, is too high. Blood glucose is the main source of energy and it comes from the food that we eat. Now, insulin, it is a hormone made by the pancreas, beta cells of the pancreas. It helps glucose from food to get into your cells, and from there it gets converted into energy. Diabetes is divided into 3 main types: type 1 diabetes, type 2 diabetes, and the gestational diabetes. So this is a simple figure which let us understand what is the normal physiology, what happens normally in the body, and what happens in type 1 diabetes and type 2 diabetes. Now, in this figure, if you consider this blue circular things are the glucose, this purple thing, this is the insulin, this green color is the insulin receptor, this yellow color is the glucose transporter. So what actually happens, as soon as we eat food, the glucose from the food, it reaches the bloodstream, and the body beta cells of the pancreas, they release insulin. This insulin, it goes and binds to the insulin receptors, which then send signal to the glucose transporter, and it uptakes the glucose from the bloodstream and then converts it into energy. This is how normally the process goes on. Whereas in type 1 diabetes, what happens? Insulin is not produced by the beta cells in the pancreas, and this is the reason the glucose that is there in the bloodstream is not removed from the bloodstream. Why? Because for these insulin receptors to get activated, we require insulin. So if insulin is not there in the bloodstream, these insulin receptors will not get activated, they will not send signal to the glucose transporter, and it will not take the glucose that is present in the bloodstream, and this leads to higher amount of glucose in the bloodstream. Whereas in type 2 diabetes, insulin is produced by the beta cells of the pancreas, and this insulin goes and bind to the insulin receptor. But the main problem is there is a defect in sending the signal from the insulin receptor to the glucose transporter, and this results, since no signal goes from insulin receptor to the glucose transporter, the glucose still remains in the bloodstream despite insulin being produced by the body. So this is type 2 diabetes. So let's just revise once again. Type 1 diabetes, in which insufficient insulin is produced, the insulin receptors, they don't get activated, and the glucose transporter do not uptake glucose from the bloodstream. Whereas in type 2 diabetes, which is also called as insulin resistance diabetes, over here the insulin is produced by the beta cells of the pancreas. The insulin, it goes and binds to the insulin receptor, but there is defective signaling. No signal goes from the insulin receptor to the glucose transporter. Thus, still the glucose level will remain high in the bloodstream. Type 1 diabetes. It is caused by the destruction of insulin producing pancreatic beta cells by an inappropriate autoimmune response. So in type 1 diabetes, the beta cells that are responsible for production of insulin in the pancreas are destroyed by autoimmune response. Now let's take a case study. In our presentation, we are going to study several case studies for each and every disease to understand the disease better, because gene therapy is mostly understood by the case studies. A study by Lee, Kim, Shin and Yoon performed in Korea. Their results were published in Nature. This experiment was performed on mice and rats, but the results may result in future implications for humans. Scientists, they used a recombinant adeno-associated virus to insert a gene that results in the expression of a single chain insulin analog into streptozotocin-induced diabetic rats and autoimmune diabetic mice. First, the gene was cloned under the L-type pyruvate kinase promoter, which regulates the expression of SIA in response to glucose levels. The LPKSIA gene was then attached to a recombinant adeno-associated virus and integrated into the host chromosomal DNA. After insertion of this gene, the rats displayed a drop in glucose levels that reached a range of normoglycemia within 1 week of treatment. The rats remained in this range for more than 8 months. That's a good result. As we can see from this diagram, we can see over here, as this represents the increased dose, and this is over a period of time the blood glucose level when it was monitored with the help of increased dose. We can see as the dose was increased, the blood glucose level over time reduced. Now, in addition to 8 months of controlled glucose levels, there were no visible side effects from the gene therapy. Another good positive response. While the results did not show permanent remission, the control of glucose levels from the insertion of SIA gene was promising. This form of gene therapy may provide a cure for type 1 diabetes for humans in the future, but a lot more research would be required before that can happen. Coming to our second example, x-scid, that is X-linked severe combined immunodeficiency. It is an inherited disorder of the immune system that occurs almost exclusively in males. Boys with X-linked SCID are prone to recurrent and persistent infections. Why? Because they lack the necessary immune cells to fight off certain bacteria, viruses, and fungi. Not clear? Let's go into detail. What's the cause of X-SCID? Mutations in the IL2RG gene that causes X-linked SCID. This gene provides instructions for making a protein that is critical for normal immune system functioning. This protein is necessary for the growth and maturation of developing immune system cells that are called as lymphocytes. So if there is mutation in this gene, what will happen? We will not get functional lymphocytes. Now imagine the damage. Lymphocytes, they defend the body against potentially harmful invaders. They make antibodies and help regulate the entire immune system. So mutations in this gene, it prevent these cells from developing and functioning normally. Without fully functional lymphocytes, the body is unable to fight off the infections. So what is the inheritance pattern? This condition is inherited in an X-linked recessive pattern. To understand in depth about the X-linked recessive pattern, please do refer my previous lectures. The gene associated with this condition is located on the X chromosome, which is one of the two sex chromosomes. As we all are aware, in males, they have only one X chromosome and other one is Y chromosome. So one altered copy of the gene in each cell is sufficient to cause the condition. Whereas in females, they have two X chromosomes. So a mutation would have to occur in both the copies of the gene to cause a disorder, and that is pretty rare. Because it is unlikely that females will have two altered copies of this gene, males are affected by X-linked recessive disorders much more frequently than females. A characteristic of X-linked inheritance is that fathers cannot pass the X-linked traits to their sons. So if we'll take a look, X-linked recessive, wherein the father is affected and the mother is unaffected. We can see over here the father cannot pass on this gene to the sons. The father will pass on this gene to the daughter, but since there is mutation only in one set of gene on this chromosome, there is another set of gene on this chromosome that will work properly if this gene is not working properly. This is a reason the females, they will only act as carrier. Whereas in the second case, the father is unaffected and the mother is a carrier. So in this case, the mother will pass on this gene, and 50% the chance the daughter will be a carrier, and 50% chance that the son will be affected. So it affects young children and is usually fatal within their first year of life. What's the treatment option? Bone marrow transplants are usually the best option for treatment. But with the difficulty in finding a donor who matches the patient, gene therapy has become a new alternative. Clinical trials for treating x-scid patients have been marked by mixed results. There's a very interesting case study to study in this x-scid. One of the patients involved in the Fisher trials have developed leukemia 2.5 years after the initial gene therapy treatment. Now, 2 of 11 patients involved in a similar study in France have also developed leukemia. This was mentioned in these journals. The gene therapy treatments have resulted in the overexpression of the gene that may be an oncogene and is located at the site of retroviral insertion. The site of insertion is the first intron of LMO2 gene, which is located on chromosome 11. But unfortunately, it is also the site of translocation that occurs in leukemia. So this observation clearly correlates the retroviral insertion as the cause of leukemia in the patients. So if we take a look, it resulted in unintended consequence. So there's a law of unintended consequence. It is still unknown whether the development of leukemia in these clinical studies was a result of a premature treatment, which could be eliminated with further research and development, or if it is a permanent risk. Despite the fact that without treatment, x-scid is a fatal disease, there is still an ethical question of whether or not it is right to subject a sick child to the possibility of developing another disease through the risk of gene therapy treatment. Though they were trying to treat x-scid, that resulted in another disease that formed in those kids, that was leukemia. So what they actually did in this gene therapy, a bacterium carrying plasmid with cloned normal human ADA gene, it was genetically inserted into genetically disabled retrovirus, and the cloned ADA gene that was incorporated into the virus, it was then infected into the T cells, and those ADA genes were then transferred to the cells. Cells were grown in culture to ensure ADA gene is active, and these genetically altered cells were reimplanted to produce ADA. Now coming to our third example, that is cystic fibrosis. Another very interesting example. Cystic fibrosis is a hereditary disease that affects the lungs and digestive system. The body produces thick and sticky mucus that can clog the lungs and obstruct the pancreas. It can be life-threatening, and people with the condition tend to have a shorter than normal lifespan. So what actually happens in cystic fibrosis? If we take a closer look and compare the airway of healthy individual with the individual who is suffering from cystic fibrosis, this is the airway of a healthy individual. We can see, it is completely clear and air can easily pass through it. Whereas in cystic fibrosis, if we take a look, oh my god, see the extent to which it is clogged with thick and sticky mucus that is clogging the airway, and it is very difficult for air to move through it. So this is a closer look at the transverse section. The smooth muscle, if you take a look, this is how it looks, the healthy individual. Whereas cystic fibrosis, oh my god, inflammation, increased mucus secretion, thickened smooth muscle. You can see the difference between the smooth muscle of healthy individual and the smooth muscle of a person suffering with cystic fibrosis. It is just clogging the airway. It is increasing the difficulty to breathe in the air. So this thick sticky mucus, it along with the lungs, it can also affect the pancreas. It can go and block the pancreatic and bile ducts. So what is responsible basically? Which mutation? The mutation on the CFTR gene, chromosome 7, that is responsible for cystic fibrosis. So a simple video to understand the difference between airway of normal individual and the cystic fibrosis. Can you see the changes in the airway of the person suffering with cystic fibrosis? Take a closer look to it. You'll understand the difference. How it is just clogging the airway. So what's the cause of cystic fibrosis? It is caused by mutation in the gene of protein named cystic fibrosis transmembrane regulator, CFTR. And what is CFTR responsible for? It regulates the movement of chloride and sodium across epithelium. It affects mainly lungs, but also pancreas, liver, and intestine. And what's the main symptom? Difficulty in breathing. It was discovered more than 10 years ago, and efforts are still underway to develop safe and effective vectors for introducing this gene into respiratory epithelium. Let's take a look at the case study. After several phase 1 clinical trial results show that mostly adenovirus vectors are used to transduce either nasal or pulmonary epithelium. Disadvantage: inefficient amount of transduced cells are transduced. Transient nature of gene expression lasts for a few days. Now, what is transduction? It is a process by which foreign DNA is introduced into a cell by means of a vector. Later, the AAV vectors for delivering CFTR are now in clinical trials. Preclinical studies demonstrated long-term transgene expression. Long-term safety and efficacy study of adeno-associated viral vector are under evaluation. Liposomal vectors are also under evaluation in cystic fibrosis. Liposomes may be mildly effective, but their activity does not last. So we'll take this example that was from the pharmaceutical journal, wherein the cationic liposome containing plasmid containing the normal CFTR gene is induced into the cell either by endocytosis or by direct fusion with the cell membrane. Through endocytosis, it reaches inside the cell, followed by escape, and the plasmid DNA enters inside the nucleus, where the normal functioning CFTR gene produces the protein, and that protein then is released from the cell. And by the direct fusion method, it directly fuses inside the cell, where the CFTR is transcribed into mRNA, which exits the nucleus and is translated to produce CFTR protein. The protein moves to the cell membrane and restore the lost function. Thank you. Please leave your questions in the comment section, and Please do subscribe to stay updated with the latest videos. This was the part 1, and stay tuned. Very soon I'm going to upload part 2, wherein we are going to further discuss in detail several other examples of potential target diseases for gene therapy.